So I try to get the task to see what is new and where are we going on the diet in hepatic encephalopathy and in cirrhotic patients. And maybe there are not so many news. And so I try to put some literature in it, and the first topic is the importance of being nourished in liver disease. And so in this regard, I had a look on the prevalence of malnutrition in these patients in the last decades. And really, I cannot see that there is much improvement, although we are continuously saying we should nourish this patient. Then when you are on the ground, when you are in the department, you really see that still we have a large percentage of these patients with malnutrition. And even more, you know that now there are these new methods which are not bedside, but for research sometimes, which are not precisely understand, and maybe we have bad cutoff. But what comes out is that by using this evaluation of sarcopenia with the CT scan, it comes out that even obese liver disease people may have, as you see on the left, may be with an increased BMI, but with a decreased muscle. So we are more sensible in some way to find out sarcopenia, and so even more now we are on the red line, and these methods even increase the percentage of patients who are diagnosed to have nutritional problems. And many studies have shown, I just picked the more new ones, like this study of Huisman, have shown that malnutrition goes together with the complication, and you see that hepatic encephalopathy is among a complication which is strongly influenced by malnutrition. And we presented in a previous ISHEN meeting this difference among malnourished and no malnourished patients, protein malnutrition, that in their history malnourished patients have higher frequency of episodes of overt hepatic encephalopathy, and at the same time at hospital admission they had more episodes of more severe degree. And this was true also for the prevalence of minimal, or as we say now, covert hepatic encephalopathy. And the protein malnutrition, so you see, was independently associated either with overt and with minimal hepatic encephalopathy in this group, although we know that infections and MELD and hyponatremia are also important factors. And the reason why nutrition, and especially muscle nutrition, is important for hepatic encephalopathy, we heard from Professor Jalan, and you see in healthy individuals the muscle is not doing so much in ammonia metabolism, but we know that when urea synthesis is impaired, the muscle has a crucial role for many reasons, both for detoxifying ammonia and also through the protein breakdown by synthesizing more glutamine that goes to the enterocyte and produce ammonia that is then staying more in the individual and cannot go in the urea cycle to be excreted in urine. What is new? There were some guidelines which were published recently, and also the hepatic encephalopathy chronic liver disease practice guidelines had a chapter with regard to nutritional recommendation, and you see that were not much news, because these daily energy intakes were those which were proposed, I can think about a decade ago, and maybe something new is that we have to modulate this when we have an obese patients, and more obese patients become cirrhotics now. And daily protein intake, also with a grade A, should not be specifically reduced in hepatic encephalopathy. And you see this knowledge that protein do not harm in hepatic encephalopathy has grown and grown also after the study of Juan Cordoba. With regard to the other options, we have a grade A, but the studies are very different one from the other. I just take this review from Professor Dasarati and ISHEN, and they put together a number of studies looking at the late evening snack, both in the short term and in the long term, but it was even difficult to have a proper meta-analysis in this regard. So you see that the short-term outcome is to avoid in this patient the rapid transition to starvation, and so to prevent utilization of lipids and proteins coming from muscle as main source of energy after an overnight fast. So you shorten the period of fast, and the nitrogen balance was also improved by these snacks, and these are a sort of other studies giving something during the night. And you see they show that nocturnal supplementation was efficacious also for total body protein, and also other studies show that this supplementation and hypercaloric diet could give a beneficial effect in patients with hepatic encephalopathy. So this study were not all together showing the same things, but many of them show an improvement in fat-free mass and change in body composition, improvement of quality of life, and decrease in blood ammonia, which could be of interest in regard of hepatic encephalopathy. And what about the branched chain, which were now introduced in many guidelines? So it's strange that branched chain amino acids were known to be low in cirrhotic patients from many, many years ago. And you know, they are essential amino acids. They are frequently low in liver cirrhosis, and they change the ratio with aromatic amino acid. And now we know better that branched chain amino acids are reduced in blood due to their action to facilitate ammonia detoxification in skeletal muscle and also in brain. So it was shown from experimental models that hyperammonemia caused branched chain amino acid reduction in animals, and that an inhibitor of glutamine synthetase could reduce leucine oxidation in these patients. This is because branched chain amino acid goes to the synthesis of glutamate, and glutamate can incorporate an ammonia for glutamine and therefore increase ammonia detoxification in muscle, dispose of muscle ammonia. At the same time, branched chain amino acid can be incorporated in the tricarboxylic cycle, and they can give a carbon atom to form alpha ketoglutarate. And from this point of view, it appears that they give one ammonia, and they can take up two ammonia to form glutamate and glutamine. However, this is not a real net balance, because as we also heard from Professor Jalan, if you don't get rid of this ammonia, you still have in the enterocyte that glutamine goes and produce ammonia once again. So probably the only way to get rid of this is to complex this ammonia in some way, so that you can have it out in urines, or maybe if you avoid glutamine breakdown, or if you can increase glutamine incorporation in protein synthesis, which could be a possible future. The interesting thing is that branched chain have so many other effects, like stimulate liver regeneration and especially leucine, can induce an hepatocyte growth factor from stellate cells. And altogether, many studies have utilized this in some way supplement to the nutrition. And it is interesting to see that rather recently, last year, a systematic review with meta-analysis appeared on a branched chain supplement, and the point of this meta-analysis were that they use individual data. And for instance, from our study, which had patients not all with hepatic encephalopathy, they kept only the individual data with a history of hepatic encephalopathy. Then the individual data of the study in Spain and in Japan were taken. And this meta-analysis showed that there was a beneficial effect in the improvement of hepatic encephalopathy manifestation, taking all together with this study, and with a difference when a study published only in abstract form was introduced. No difference was shown for mortality. In the same guidelines, you know that with regard to the use of branched chain amino acid enriched solution for hepatic encephalopathy, we still deal with very old studies, and we know that, as we hear today, there are so many problems of interfering factors when you have a severe hepatic encephalopathy and precipitating cause that we have no recommendation about using this substance in the acute episode. So where are we going, if we know? And maybe we heard a lot in this Congress about the great microbiome or microbiota, and about, an idea could be to modulate this microbiota through the diet. We already heard that this could be modulated through probiotics from Professor Sharma. We heard in this Congress that gut microbiota is different from normals, that we have lower gene richness, and some species are more abundant and some species have a lower abundance. And the species with lower abundance are the good ones, for instance, those who produce butyrate, and this changes somehow goes together with the severity of liver disease, as you see, the MELD score, the Child-Pugh class, and we had a lot of data in this regard from Professor Jasmine Bajaj, and showing that there are some species which are more in some way more prevalent in healthy individuals with respect to cirrhosis, and interestingly showing specific data in cirrhotic patients with and without hepatic encephalopathy. And you see that sometimes they have an even deeper decrease of some species like Ruminococcus, and some further increase in some species are associated with poor cognition in individual tests. So, but it was right to say that we should say what these species do, and so the functional role of this changed gut microbiome. And the authors in Nature, they say that in liver cirrhosis there are many associated markers regarding ammonia, GABA biosynthesis, and manganese, and they propose that, for instance, that manganese-related transport system and the GABA biosynthesis module may be involved in the hepatic encephalopathy, and microbiome modulation may therefore act as a therapeutic option in the treatment of this disease. So how can we modify intestinal microbiome? We heard before prebiotics, probiotics, and symbiotics. And with regard to the diet, we can stay in here, in prebiotics, because you see that substances contained in food that reach the colon can change the bacterial proportions. And among them, fibers, for instance, are very important because they undergo bacterial fermentation and lead to production of many substances which we heard in the first day are very important for the colonocyte health. And I just report you some idea which are in the literature of nutraceutical products, and the idea of using products which are modified from the diet and are not really specific therapies or drugs. For instance, you see they're studying human milk oligosaccharides. In human milk, there are these oligosaccharides which are potent bioactive molecules able to enhance bifidobacterium lactobacilli, reduce harmful bacteria, and these oligosaccharides have a specific role because they can stop the bacteria to go inside with different ways. One way is to mimic the receptor, and another way is to go into the cell and to produce an altered glycan-related gene expression, producing a different receptor so that the bacteria cannot go, some bacteria cannot go inside the enterocyte barrier. These oligosaccharides are present but in very low trace in the bovine milk. So they are trying to concentrate these products using a serum, a hypermate, which is processed and can have high concentration of this oligosaccharide and may represent an alternative to give an organism the same opportunity that the human milk. But there are also other ideas. For instance, in this study, they produce milk from transgenic goats expressing lysozyme at very high concentration in their milk. And so pigs were fed with these two different milk, and gut microbiota was evaluated after 2 weeks. And you see this class were those decreasing, and this class were those decreasing, and many bacteria changed after the assumption of this specific transgenic produced milk with higher concentration of lysozyme. And also you know that vegetarian diet, due to the high fiber content, can modulate gut microbiota, and vegetarian diet have been supported for their use in hepatic encephalopathy. And there was a very recent paper in Nature just dealing with plant-based diet and animal-based diet, and you see the difference in fiber in the plant-based diet and low fiber in the animal diet, the difference in fat, and some difference also in the protein intake. So take care that a vegetable plant-based diet can be have less protein. And these authors reported all sorts of changes that can occur with the animal diet regarding a lot of groups of bacteria and different classes which were modified by using an animal diet or could be modified on both diets. So we have a lot of opportunity when we go inside the literature which is maybe already present to define better which are the way we need to go. And this was in Journal of Clinical Nutrition, just to compare vegetarian and vegan diet, so even more strict diet, and you see that coli were sharply decreased, and Enterobacteriaceae as well, the more the diet was restricted to vegetables. Of course, the support of proteins can be a problem then. So we know that the study on vegetarian diets are very few and old, and associated treatments were a lot, so probably this can be a further program for the future. And this was shown by Professor Sharma, the meta-analysis showing that gut flora modulation may obtain good results with probiotics, so maybe we will obtain in the future good results and less expensive also by using modified foods for these patients. And thank you for your attention.